Hello friends, let us discuss today about genetic disorder and specifically Mendelian disorders. Now, why they are termed as Mendelian disorder? Because mutation or alteration is happening within single gene. So they are genetical disorder or Mendelian disorders. So here disorders are caused by alteration or mutation of a single gene. Now the pattern of inheritance of Mendelian disorder can be traced by family pedigree analysis. Can be dominant or recessive by their nature, and by the pedigree analysis it is easy to understand whether the trait is dominant or recessive. We have already covered the pedigree analysis mechanism into previous lecture. So today we are going to discuss all the Mendelian disorders one by one, and the first disorder is hemophilia. So hemophilia is the disease which can be which can affect an individual by not having a blood clotting. So it is abnormality of blood clotting mechanism. So the person who is affecting, even if a simple cut he observe or she observe, it can result into non-stop bleeding. Ready? So blood is not getting clotted. That is the effect which is seen in affected person. Now what is hemophilia? It is sex-linked recessive disease. Now how to go through or how to identify whether it is recessive or dominant, and how to identify whether it is autosome or sex-linked, we already have covered. So straightway we are discussing about the type of disease right now. So it is sex-linked recessive disease, and in this a protein involved into the blood clotting is affected. A simple cut also can be resulted into non-stop bleeding, and it is controlled by a pair of allele, that is capital H and small h, where capital H is normal and small h is responsible for hemophilic condition. Ready? So the condition is caused by a recessive gene, that is small h. So please take the screenshot. Fine. Here the entire mechanism is explained about hemophilia. The heterozygous female which is carrier, whether it is X-linked, always heterozygous female are termed as carrier. You are supposed to remember that. So heterozygous female are carrier for hemophilia. It may transmit the disease to their sons, right? So in the case which is given into the diagram, second case female carrier and normal male. So it is X-linked character, right? It is the X chromosome with mutation. So that X chromosome is given to male, and from father it is getting Y chromosome. So it is a 50% possibility that a male child of these pair, this couple is getting the hemophilia. Ready? So it is the possibility for this couple to have 50% of chance to have the male child with the hemophilia. You can easily conclude by the diagram. The possibility of female becoming hemophilic is very rare. Why? Because it is X-linked recessive trait. So only in the case where two recessive genes are coming together, that is recessive homozygous condition, if found into the female, then and then only it is a chance of getting female which is affected by hemophilia. So possibility of female becoming hemophilic very rare, because mother has to be at least carrier and father should be hemophilic. So if mother is carrier and father is hemophilic, these condition only can be resulted into hemophilic female. Ready? Queen Victoria is a classic example, right, of the carrier of this hemophilic disease, so that her family pedigree shows a number of hemophilic descendant. So that's the first example, which is actually showing entire pedigree or mechanism of spreading of the hemophilia. Okay, if you see condition number 1, here normal female and hemophilic male are there. So if this condition is there, X chromosome from normal female will be given to each and every individual. But X chromosome is given to female only from the father, which will be resulting into carrier daughters. So all the daughters will be carrier if father is affected. If father is hemophilic, mother is normal, then all the resulting female progeny, female offspring, female daughters are going to be carrier. That's to be noted, points to be noted. If female is carrier, father is normal, then possibility we already have discussed that 50% of the chance to have their son to be hemophilic. Ready? And if father is hemophilic and mother is carrier, then they have they have chance of getting hemophilic female as well. So these are the possibilities of getting hemophilia. Please take the screenshot of this slide. Second disorder is sickle cell anemia. Now, according or from the name only you can judge the abnormality or disease. Here blood cells are appeared like sickle, and that is why the abnormality is termed as sickle cell anemia. Now why it is anemia? Anemia is yellowing the blood. So blood turns to be yellow, then the red. Now if the RBC are turning into sickle, shaped, it is definitely some kind of denaturation which is happening there, and our blood is red because of the RBC and hemoglobin which is present on the RBC, right. This disease is actually related with the production or quality of RBC. How? That is what we are going to study now. First, what kind of disease it is? It is autosomal and recessive trait, and it can be transmitted from parents to the offspring when both the parents are carrier for the genes. This is the only requirement, when both parents are carrier for the genes or heterozygous, or definitely if they are anemic, then it is 100% chance to forward the disease from one generation to the other. But even if they are carrier, that is a possibility of getting spread into the next offspring. The disease is controlled by pair of gene HbA and HbS. Now, there HbA is normal and HbS is, S stands for sickle cell anemia. So, HbS is mutant gene. So, homologous dominant condition HbA is normal. Now, heterozygous condition HbA-HbS represents carrier of the sickle cell trait, and homozygous recessive condition, that is HbS-HbS, is showing the affected person, right. It is having anemia. So, anemia is nothing but dealing with the oxygen carrying capacity. It is a reduction in oxygen carrying capacity due to abnormal structure of RBC, right. Please take the screenshot. Now, this disease is caused by the substitution of glutamic acid by valine at 6th position of beta globin chain of hemoglobin. Okay. Let us first discuss about the structure of hemoglobin. Now, as we know that hemoglobin is having 4 polypeptide chain, 2 alpha polypeptide and 2 beta polypeptide. Production of alpha polypeptide is controlled by 2 different genes, and production of beta polypeptide is controlled by a single gene, which is responsible for creation of beta globin. Now, if the gene which is responsible for the production of these beta polypeptide, that is beta globin, that gene is affected or mutant, then there is a chance of getting sickle cell anemia. Ready? Now, how it is getting mutated? If you see the diagram which is given here, which is similar to the diagram which is into the textbook. So this is normal HbA gene, where the genetical code, it is a sixth position, sixth codon, which is responsible of the gene which is responsible for synthesis of beta globin. It shows CTC, right? A codon, as we know that it is having a 3 base pair. Now this is dominant gene, so it is going to produce your hemoglobin, beta globin actually. So when it is transcripted, so mRNA which will be produced, so CTC is going to be GAG, and that GAG is code for glutamic acid. So 6th number amino acid will be glutamic acid. So this is a normal process. But if it is HbS gene, that is mutant gene, T of the middle is replaced by alanine, that is A. So coding is, DNA coding is CAC. So transcription or mRNA will be showing GUG, and that will be responsible for the production of the valine at the 6th position. So this entire protein is no more responsible to serve as beta polypeptide. It is slightly different protein than the normal beta globin. So this is how these trait is controlled or altered, with what we can say. So this is due to a single base pair substitution at 6th codon of the beta globin gene, that is GAG to GUG, what we have seen just before. So the mutation or mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in shape of RBC from biconcave disc to sickle cell structure. Now due to that, what happens is that its oxygen carrying capacity reduces. That is the reason why the appearance, which is supposed to be red, must not be seen. That is why the color of blood plasma, which is slightly yellow, is more highlighted or is appeared more. So redness will be disappeared from the blood, and the blood appeared as yellow, and this is again, it is replacement is shown. Glutamic acid is replaced by valine. I request you to take the screenshot of this slide. Okay. The third slide, the third disease. Now, if you see third disease, it is something abnormally represented, phenylketonuria, right? Now, as far as the disease is concerned, it is known as inborn error of metabolism. Now, why it is indicated like inborn error of metabolism? Because it is, this disease is seen on the genes which is responsible for the production of the enzyme, right? So here enzyme producing gene is getting mutant. So, mutation is resulted into the gene which is responsible for a production of a particular type of enzyme. So, that particular enzyme if is not getting produced, it is resulting into the error in a particular type of metabolic reaction, and that is why it is said to be inborn error of metabolism, because that gene is not functioning since the child is getting birth. That is why inborn error of the metabolism, right. So, that is the meaning of the name which is given to them. Now, let us see what this disease is. First, the type, it is autosomal and recessive trait. It is caused by mutation into the gene that codes for enzyme named phenylalanine hydroxylase, and it is a single gene mutation. So, what happens is here is that enzyme which is coding for phenylalanine hydroxylase is not getting produced, or it is ruptured. So, affected individual lacks the enzymes, right. So, affected individual lacks the enzymes. So, they are not converting phenylalanine into tyrosine, because this enzyme is responsible for this reaction, which is not going to happen. Ready? In that condition, what is the result? So, phenylalanine is getting accumulated and convert into phenylpyruvic acid, and its derivatives, right. So, phenylpyruvic acid remains into the blood for the longer period of time, accumulates at various parts of the body including brain, and if they accumulate into the brain, resulting into the mental retardation, also create huge distribution around the body by blood. So, they are also getting poor absorption by the kidney as well. Ready? So, they are continuously getting excreted through urine. So, urine has a large quantity of phenylketone, and that is why the disease is named phenylketonuria. In excretion, you find huge quantity of this phenylpyruvic acid. So, acidic urine is found, right. So, here is the diagram which is showing that. So, dietary protein is converted into phenylalanine or tyrosine. Whatsoever phenylalanine is produced, right, has to be converted into tyrosine. So, this reaction has to go this way, because we need these, and responsible for the metabolic reaction which is converting phenylalanine to tyrosine. This enzyme, that is phenylalanine dehydroxylase, is mutant. The gene which is producing these is mutant, so it is not getting produced. So, a reaction of phenylalanine, which is in turn to be converted into tyrosine, is not getting done, and this phenylalanine is actually forming the phenylpyruvic acid, and that is accumulated into the brain to produce retardation. So, that is the condition known as phenylketonuria, right. this is excreted largely by urine. So, in urine you can find huge debris of the pyruvic acid. So, that is why phenylketonuria is the name of the disease. Why inborn error of the metabolism? Because it is concerned, or it is found that the gene which is responsible for production of a certain enzyme is found to be ruptured or mutated. So, that is what you are supposed to remember as well. Please take the screenshot. Now, the disease named thalassemia. Now, thalassemia is again the disease which is related to the blood, right. So, it is blood disease. It is autosomal recessive disease. Now, transmitted from the parent to the offspring when both the parent are unaffected carrier, right. Recessive disease always shows this character or pattern. So, both when they are heterozygous, it can be transmitted to their offsprings. It is a possibility, right. Now, if both of them are affected, then 100% it is not a chance, but it is 100% it is getting forwarded from parents to the offspring. Now, it is due to mutation, specifically deletion. It result in, look, you are supposed to remember the word reduced synthesis of one of the globin chain, ready, from alpha and beta chains that make up the hemoglobin, and that is why it forms abnormal hemoglobin molecule which is resulting into anemia. Now, thing is that it is not sickle cell anemia, it is far away different than sickle cell anemia. In what manner? Look, thalassemia differs from sickle cell anemia because thalassemia is quantitative problem. Why quantitative problem? Because quantity of hemoglobin is reduced, which is actually highlighted here into discussion, ready. So, quantity of hemoglobin is getting reduced, number of hemoglobin which is produced in your body is reduced. So, it is quantitative disease, whereas sickle cell anemia is qualitative problem because it is synthesis of incorrect function of the globin. So, carrying capacity of a single RBC reduces, here number of RBC reduces, ready. So, that way they are different than each other, what you are supposed to remember. Now, we should follow, or we should think about how the thalassemia works, which are the possible mechanism responsible for spreading of this disease. You take the screenshot first. Fine. There are two types of thalassemia. Number one, alpha thalassemia. Number two, beta thalassemia. If thalassemic gene is referred to the production of alpha globin chain, which is caused by HbA1 and HbA2 genes, then it is alpha thalassemia, and production of beta globin is reduced, then it is called beta thalassemia. So here production of alpha globin and their production of beta chain is affected. Now responsible for the alpha thalassemia are HbA1 gene and HbA2 gene, whereas responsible for production of beta chain are HbB single gene. HbA1 and HbA2 are found on chromosome number 16, whereas beta thalassemia, the gene responsible for beta thalassemia, that is HbB gene, is found on chromosome number 11. So either mutation is found on chromosome number 16, then it is alpha thalassemia, or if mutation is found on chromosome number 11, then it is beta thalassemia. So this is alpha thalassemia is due to the mutation or deletion of one or more the 4 genes, 2 pair of genes, that is 4 genes. The more genes are affected, the less alpha globin molecules can be produced. So that is why it is quantitative disease than the quality. Ready? And it occurs, beta thalassemia occurs due to the mutation of one or both the genes. So 1 or 2, again quantitative. So thalassemia occurred due to either HbA or HbB genes mutation, and accordingly they are termed as either alpha thalassemia or beta thalassemia. Please take the screenshot of the slide. So that's where I am concluding and ending the all Mendelian disorders. We will be continuing with the chromosomal disorder into the last lecture of our discussion of chapter number 5. Till then, have a nice time. Bye and take care.